Hi, I'm Jamie Batch, your instructor for AMP2. We're going to continue on with our discussion on the male reproductive system. We just finished talking about the overall anatomy of the scrotum and testes, and now we are going to, sorry, I lost my screen there. Now we're going to continue with how sperm are formed in the process called spermatogenesis. So it involves the process of mitosis and meiosis. There's actually three main ingredients that are taking place in spermatogenesis. So the first thing that has to happen is mitosis, and this is just the general process of cell division where two identical daughter cells are formed. In this process, the new cells that are created have two full sets of chromosomes. This is happening in the seminiferous tubules, and this is happening with a special type of cell called a stem cell. They're called spermatogonia, and these spermatogonia will divide and they will create two identical cells. One of them stays behind and remains the spermatogonia so it can duplicate itself some more, and the other one is then going to go on and divide some more to become a sperm cell. So here you see the general process of this stem cell. In this case, we're talking about sperm. It's called a spermatogonia. The spermatogonia is going to divide into two, creating another stem cell, and then the second cell that is produced from that division is going to be the cell that goes into the meiotic process to become a sperm. So let's take a look at the next step. The next step is called meiosis. So that second cell from that first mitotic process is now going to undergo meiosis, or sometimes it's pronounced meiosis. It's potato, potato. It doesn't matter. This is a special type of cell division in which instead of two identical daughter cells being created, it's actually four half cells that are being created. So it's a series of two cell divisions that are reductive in division. So instead of creating two cells that have 23 pairs of chromosomes, we're creating two cells that just have 23 chromosomes. The chromosomes aren't paired anymore, because the whole point here is reproduction. So we want to get cells that have half the number of chromosomes that they need so they can combine with another cell that has half the number and come together through the process of fertilization and create a whole cell that has all the chromosomes that it needs. So in this case, we're producing cells that are called haploid cells. Haploid cells have 23 individual chromosomes as opposed to the cells that are produced during mitosis, which are called diploid or diploid, and they have 23 pairs of chromosomes. A process called synapsis occurs during meiosis, and this is where the chromosomes will actually come together, the matching chromosomes will come together and they'll form something called a tetrad, and they will actually exchange genetic parts. They will switch up the genes a little bit. This slide here is not showing you that at all. I don't know why that slide was not included, but I'll go back here for a second. So during this process of synapsis, which occurs during meiosis, the chromosomes, which are identical, will cross over each other, pair up, and exchange parts. This is why we're all a little bit different. It's because of this process called synapsis that occurs during meiosis, and it's just a genetic recombination. It's a reshuffling that occurs. So this is meiosis, and I encourage you to flip back to 2 slides to look at the process of mitosis and see how these processes are different. So in meiosis, you start with a normal cell that has 23 pairs of chromosomes. During meiosis 1, you have these tetrads that form up. These chromosomes that are identical will cross over each other and switch parts. This genetic recombination, typically what you would see is kind of a, I don't know, different colors here. Either way. Then these two cells that are created from the first division do not duplicate their DNA, and so the chromosomes are then cut in half, and we end up with 4 half cells, or haploid cells at the end of meiosis. So the third step of spermatogenesis is called spermiogenesis, and this is actually getting our sperm to mature into fully functional spermatozoa. So this does not occur in the seminal vesicles. This is going to, in the seminiferous tubules. This occurs later on in the pathway, so to speak. So let's talk about this mitosis. So the spermatogonia or spermatogonium, which are those stem cells, they're located in the seminiferous tubules. They're actually located on the outer rim of the seminiferous tubules. And as the mitotic process is occurring, the cells are getting pushed towards the middle of the tubule or towards the lumen of the tubule. So by the time the cells undergo their meiosis, undergo their reproductive divisions, they're in the lumen of the tubules and can be swept off towards the rete testes and into the epididymis for ejaculation to occur and spermiogenesis. So as they go through mitosis, the first step, they're on this outer ring, those stem cells, the spermatogonium, undergoing mitosis on the outer ring. They're going to divide and form two daughter cells. One is going to remain there so that it can divide again and form more sperm. The other one is going to go on and undergo meiosis. This other cell is called a primary spermatocyte. The cell that goes on to undergo meiosis is called a primary spermatocyte. It takes approximately 16 days for that spermatogonium to become a primary spermatocyte. So this process is relatively slow, which is crazy because men are producing sperm at a very rapid rate, but it is a rather lengthy process because it's about perfection, right? These cells have to be perfect because they're going to make an offspring, and you don't want to have an offspring that's doesn't have a correct set of chromosomes. So after we have mitosis, our primary spermatocyte is going to undergo these reductive divisions of meiosis. In meiosis 1, which is the first division, this primary spermatocyte, which has 46 chromosomes, divides into two cells, each containing 23 chromosomes. These 23 chromosomes are duplicated and will separate in the process of meiosis 2. So meiosis 2, our secondary spermatocytes divide to produce the haploid spermatids. These spermatids contain non-duplicated chromosomes because they split, right? And each primary spermatocyte produces 4 spermatids, and this is a relatively quick process. Finally, the last process is spermiogenesis, which is the physical maturation of these sperm. This is getting them ready to move, getting their flagella, ready to propel them, etc. And this can take a while as well. You don't have to know the time frame. It's just kind of as a reference. So this slide basically summarizes everything. Let's break it down one by one. You have your mitosis that takes place, your spermatogonium dividing into another spermatogonium and a primary spermatocyte that is diploid. Then in meiosis 1, you have DNA replication occurring. Your primary spermatocyte is undergoing synapsis, this kind of crossing over that occurs, mixing up the genes, dividing into secondary spermatocytes. Our secondary spermatocytes in meiosis 2 do not duplicate chromosomes, they just divide. And now we have haploid spermatids. These haploid spermatids will undergo spermiogenesis to form all of the parts of a functional sperm. This is a picture of a functional sperm. I'm sure you've seen one of these at some point in your life. So you know that a sperm doesn't look like a typical cell that we're used to seeing. So there are some characteristics that make it special. They lack most of their organelles that most cells have, and they actually lack most intracellular structures as well. They want to be nice and light so they can move and swim super fast to get into that female reproductive tract. So they have something called an acrosomal cap or an acrosome. This is basically a little hat, so to speak, on the head of the sperm, and it contains enzymes that are going to help that sperm crack the egg, so to speak, and get into the egg. It's going to allow the sperm to eat away at the outer layer of the oocyte or egg cell so that fertilization can occur. The head is where the chromosomes and nuclei are located. There is a neck that has centrioles, and those centrioles hold in place microtubules. The microtubules extend through the middle piece and the tail, and that is going to allow the sperm to swim. The middle piece has the mitochondria, which is making the ATP that's needed for that whip-like motion of the tail. There you see acrosome head, neck, middle piece, and tail. You can pause the video here, go back and review this section. We're going to keep going, talking more specifically about the seminiferous tubules here. As we've mentioned before, they're very tightly coiled tubules within the lobules of the testes, and this is where sperm are formed. This is the site of sperm formation. The spermatogenesis and spermiogenesis takes approximately 9 weeks from the time we have a spermatogonium dividing to the time that we have a mature sperm cell. So each segment of our seminiferous tubule is going to basically represent a different phase of spermatogenesis. Every tubule is continuously producing spermatozoa. Here you see those highly coiled tubes, and if you can imagine, these kind of dark purple areas are the tubules themselves. So the outer dark purple rim is where their spermatogonium are, and as those dark purple cells move closer and closer towards the light pink area in the middle, that's that process of mitosis and meiosis that's occurring. So those are different spermatocytes at different stages of their development. And then the pink stuff in the middle, those are the spermatozoa. They're going to be basically pushed along through the seminiferous tubules into the straight tubule, the rete testes, the epididymis. So the seminiferous tubules are surrounded by lots of connective tissue. There are spaces between the tubules that contain blood vessels and areolar connective tissue, as well as interstitial cells, which are also called Leydig cells. These cells are there to secrete hormones like androgens and testosterone, basically to fuel sperm development. The spermatogonium themselves make up the wall of the seminiferous tubules. As I said, that outer edge is the spermatogonium, and then spermatocytes, and then spermatids that will develop into spermatozoa. There's also nurse cells within the seminiferous tubules. These are special cells that are there, again, to help protect and encourage the maturation of these cells. And then spermiation is the process in which the spermatozoa are basically detaching from the nurse cells and being let go into the lumen of the tubule to head towards the epididymis. So those nurse cells are actually joined to the spermatozoa by very tight junctions. They form this barrier, and there's a basal compartment and a luminal compartment. The basal compartment is where the spermatogonia are located, and the luminal compartment is where that spermatogenesis is occurring and meiosis. So think of the nurse cells as kind of like this net of cells that the spermatocytes are passing through as they're making their way from the outer edge of the tubule into the lumen of the tubule itself. Here you can see them a little bit better. So you can see here's a nurse cell, and here's a nurse cell, those big kind of blobs in the middle holding everything together. It's the purple stuff, right? So all of these cells are embedded in a nurse cell, and the outer edge makes up that spermatogonium. And as those little blue cells get pushed towards the lumen of our seminiferous tubule, they're undergoing the mitosis and meiosis to form fully functional sperm. So that whole purple thing is the nurse cell. The nurse cell is kind of the meat of our seminiferous tubules, and these spermatogonium, primary and secondary spermatocytes, are kind of stuck in the net of these nurse cells, so to speak. Kind of a cool thing. I'm actually going to stop this video here because I have to go do other things, but I will be back with another video, and we can talk about how those sperm become activated and then move on to the female reproductive tract. See ya.